Hello, everyone. Thank you for joining our webinar, The Innovation Behind SkinPen Precision and Why It Matters to Your Practice. Today, we will take a deep dive into the engineering of SkinPen Precision, the first FDA-cleared microneedling device in the world, U.S. engineered and manufactured by Crown Aesthetics. We hope by sharing this information, you are able to perform SkinPen procedures on a new level in your clinic, driving both enhanced safety and efficacy for your patients. My name is Angela McDonald, and I am the Senior Director of Education and National Accounts for Crown Aesthetics. I will be moderating today's webinar. Our webinar is scheduled for 1 hour. Before we begin, let me share a few housekeeping notes. During the webinar, all lines will be muted to avoid any background noise. If you would like to ask a question, you can do so at any time during the presentation by typing your question into the chat box at the bottom right of your screen. We will conduct the Q&A at the end of Dr. Hitchcock's presentation. For those of you today who are viewing this webinar on a computer, you should see both a full screen slide presentation and at the same time a thumbnail view of Dr. Hitchcock presenting in front of a screen once we get started. You should have the ability to go full screen with either view and toggle between the two as you see fit. Please note there are portions of this presentation that do contain video. To properly view the video, you will want to go full screen with the slide presentation. For those of you today who are viewing on a cell phone, you will see either Dr. Hitchcock presenting or the slide presentation once we get started. To toggle between the two, simply swipe right. You will also want to ensure you are on the slide presentation screen at the time of the videos in the latter portion of the presentation. So let's get started. We are thrilled today to present Dr. Thomas Hitchcock, Chief Science Officer of Crown Laboratories. Dr. Hitchcock is a formally trained geneticist and tissue engineer, holding a PhD in molecular genetics from Clemson University. Dr. Hitchcock completed his postdoctoral training in tissue engineering and regenerative medicine at Duke University and vascular biology and therapeutics at Yale University, where he was involved in groundbreaking research and development of engineered human cardiovascular and pulmonary tissues. Dr. Hitchcock has more than 15 years of research experience across several therapeutic areas, including cardiology, oncology, gene therapy, plastic facial plastic surgery, dermatology, and aesthetic medicine. Most notably for us, Dr. Hitchcock has been an integral part of the Crown Aesthetics, formerly Bellis Medical team, leading our clinical and medical affairs since the launch of SkinPen in 2013. To be led by a scientist of Dr. Hitchcock's caliber is one of the many differentiating factors of our organization, which ultimately has led to our success in becoming not only the first FDA-cleared microneedling device in the world, but also currently the #1 microneedling technology in the US. Dr. Hitchcock has overseen the advancement of SkinPen and its related protocols and education through 4 device generations, most notably the development of SkinPen Precision, which you will learn more about today. Dr. Hitchcock?
Hello, everyone. I'm going to get my video up here for you. Thank you, Angela. I appreciate the intro, and thank you all for taking your time today to listen to my thoughts on the innovation that went behind the engineering of the SkinPen Precision device. If you're listening to this right now, then you've probably been given some speaking points about the quality or the degree of work and validation that went into the SkinPen Precision to make it the world's first device of its kind that the FDA cleared as safe and effective. However, I thought it would be more interesting to show you, not just tell you about the quality, design, and engineering behind the device. What we're going to discuss today and show you today is some of the concept work, the proof of concept work, that our scientists and collaborators have done that makes the concepts a little more tangible for those of you who might find some of these ideas a little more nebulous. So in this 2-part series, we'll be looking at different design elements of the precision device and show you some of the physical evidence that we've designed of why we designed the device the way that we did. Now, I want to reiterate that this is an encore presentation. We've done this before, so I want to make clear that this is not part 2 because we're planning to do a 2-part series, and this is the first of that 2-part series. During today's series, we're going to talk about the design elements that went into the disposable portion of the device, the advanced cartridge unit. Next portion, we'll talk about the more on the actual handheld portion of the device. So as Angela mentioned, I have the slides behind me here. I'm going to try and face forward because I was told that you cannot hear me properly when I turn around to point. So I'll try to do a weatherman thing and point this way. However, also remember that you can look at the slides close up if you need to by toggling. So I'll go ahead and begin. Okay. So if you see this chart here, you'll probably, if you already have a precision device or you've talked to any of the salespeople at Crown Aesthetics, you've probably seen this type of slide where we're looking at the different validation studies that were done to get the clearance through the FDA and other regulatory bodies. While we do like to make mention of this because we are very proud of this work, sometimes these things are a little nebulous. So people look at it and they say, okay, needle durability. What does that mean? How did you test for that? And you know, what can I look for needle durability? The reciprocating motion. So it says here the purpose was to verify the needle reciprocating motion meets safety specifications. Okay, well, what does that mean? How does that translate to benefit for my practice? How is it different from other devices that are on the market? Needle penetration depth. So, you know, all the devices on the market basically say that you can dial them up or dial them down to specific depths. And so how is that different from the SkinPen Precision? Why is it that we are talking about that as a validation study? And what does that mean to my practice? On this next slide here, you can see that there's 3 more here that we will be looking at today, which is the fluid ingress prevention. And so that is one of those things that is one of the top priorities for most practices, which is how do I know that when I'm doing a microneedling procedure that I'm not getting any of the fluid from the contact of the patient, like blood or lymph or plasma, sucked into the device, and so that I don't have to feel like if I don't actually clean it from use to use, I'm going to potentially cause cross-contamination. While there is, and then the Santa Claus wiping cleaning validation. So basically, these are 3 steps to ensure that your device is always sterile. But the key point is that if the first step, the fluid ingress prevention, actually works properly, then you wouldn't have to take additional steps. But some of you are more cautious than others. So there are 3 ways to basically mitigate any possible risk. And that is by using a certain disinfectant to actually wipe down the device, the biosheath to prevent any contact, even indirectly, with bodily fluids, and then the fluid ingress prevention. Now, there are quite a few companies that basically take these same speaking points. However, we have always gone a step further, and we actually will show you how we've done this. So this again is something that you can find in any of our marketing literature. This is not, I'm not going to belabor this point, but this looks to the needle durability. This basically is looking at one of the smallest components of the needling device, the needle itself, the microneedle itself, and showing that there are differences, that just because a device claims that it has microneedles does not mean that they are all the same, even down to the smallest component. So here you can see on the left-hand side here, the SkinPen needle. So these are our needles that we quality control to be all the same. We make sure that they're completely straight, that there's no imperfections that would lead to any type of tearing or anything of that nature. In the middle and then on the right-hand side here, you can see 2 other companies' devices, not our device. And these are devices on the market that you can see there are some either irregularities or the needles themselves aren't truly needles, but actually serrated blades. And so we need to just keep in mind that even the smallest components, just because somebody uses the moniker microneedling, does not mean that all of these devices are the same. Here you can see it's not even just the needles themselves, but it is the way that the needles are actually inserted into the device. This particular example is not our device again, but it is a device that you can see that most of the needles are placed at the appropriate depth or the same consistent depth, but there's one needle right in the middle that is actually significantly higher than the rest. And so I'm not sure if this was intentional by that device, but when you are actually trying to do a prescriptive type of treatment where you're looking to hit certain depth of tissue, You got to remember these are going hundreds of reciprocations per second, and so therefore that one needle will be producing hundreds to thousands of channels at that depth, where the other needles will be producing channels at a different depth. So this is just something to say that, again, even the smallest components, but not just the smallest components, but the way that their quality, the quality control for actually putting those components together truly matter. Okay, so this slide here is a image of the patent that we have been issued. So the SkinPen Precision is a patented device, and this schematic here on the left or the right-hand side here is actually a representation of the disposable cartridge, the advanced cartridge unit. And this is actually the part that is patented. So the reason why it's so different and we were able to receive this patent is because we actually did something that was quite unique, and we took the reciprocating mechanism, and we'll talk about what that is, how that's put together, etc. We moved it from the hand unit and we placed it into the disposable part. Now, there's a few reasons that we do that. One is so that because the reciprocating mechanism is the one that has the most moving parts and the most delicate parts. So this way you basically have a new fresh device every time you do a procedure. But that's just the beginning of why we did that. There's other reasons as well. We'll talk about that in the next few slides. So if you look at the next, particularly the next slide here, we have a SkinPen advanced cartridge unit, and here's one in my hand right here. We basically broke it into pieces. So we took it apart, and we put each of the pieces and the components here on the top left-hand side. And you can see this is a Scotch yoke mechanism. So basically what a Scotch yoke mechanism is meant to do is to turn rotational motion into linear motion. So the areas that are populating here, I'll go ahead and populate them all until this animation. So basically what we can see here is that there's a rotate. Now this is, the animation here is not our device. It's basically a representation of the type of mechanism that turns that rotational motion into linear motion, which is what ours does on a smaller scale. So you can see we have this, the crank, which is the rotational mechanism. And you can see that little gray dot in the middle of the crank is a pin that actually is slightly offset, so that every time it turns, there's a slotted link that is connected to the pistons and the piston plate, which has the needles in it. And that actually allows it to turn into linear motion. That slot actually, because of the offset of the needle, as it rotates, slides along the slot and the slotted yoke, and then that actually converts it to linear motion. And so if you go a little further, you can see the animation, or I'm sorry, the video here of the SkinPen. So let's go there. You can see the SkinPen actually in motion. If you look really closely at the middle there, we've actually did a cross section here. So in a normal device, you would not be able to see this, but if you look closely in the middle there, you can see the actual Scotch yoke mechanism working. Now, why is this important? Because other devices may use a similar mechanism, but the point is that they actually have that mechanism inside the handpiece and not the disposable part. And that's a very important consideration that we looked at when we were designing this pen, because what we have here is actually a coupled mechanism. So all of the rotational energy that goes into making it linear all happens in the disposable itself. So it's all coupled. That means that every rotation, the same force is pushing the needles into the skin as is the force taking it back out of the skin. So if you look at the next slide here, I have a schematic that I drew myself to demonstrate this. So if you look on the left-hand side here, this is an example of coupled retraction, and this is what the SkinPen Precision has, and this is what's unique, because ours is the only device that I'm aware of on the market that actually has this type of reciprocating mechanism where it's coupled. And that means that for every turn, and as you can see the circle on the bottom, the little oval, that's the representation of the crank. And that is in the disposable portion. So the motor turns the crank, and the crank actually causes the linear motion through the shaft there that is attached to the needles. And so the force of the crank actually translates directly to the force of the shaft here. On the return, which is represented by the red arrows, on the return of that mechanism, you see that it's the same force from the same crank and it's the same force pulling the needles back out of the skin. Because of this, you actually have on each reciprocation, you're going to have the same force pushing the needles in and pulling them back out. So every time it's going to be calibrated correctly to penetrate the skin at the depth that is dialed into the needle setting, the setting that is dialed into the needle. Now, we're going to look at some proof that shows that in a bit. So this is really the theoretical portion of our discussion, and then we'll show you the actual hands-on portion that will give you a little more sense as to the proof of concept. So if you look at the right-hand side here, this is the majority of other devices. And so this was actually the same mechanism that we used in previous iterations of the SkinPen device, and that we've moved away from intentionally, and I'll tell you why in a second. So if you look at the right-hand side here, you can see that they have also a Scotch yoke mechanism. The difference here is that the actual part where the piston is connected to the needles is uncoupled. And what that means is that the crank is connected to a piston just like with our device, the SkinPen Precision, but the difference is that now you have a disconnection between the cartridge, where you have a needle head that is connected to a piston that is disconnected to the piston in the device. So as the crank rotates, the force of the piston pushes up and then pushes the piston in the cartridge up as well with the same force. The difference now is that the way that it's pulling back is not through a direct retraction, meaning it's not coupled. So the piston in the device pulls back, as you can see in the middle part of that schematic. But the pulling back from the needle head is actually done through springs or rubber or some sort of spring-like mechanism. That's why we're calling it a spring-modulated retraction. So the force is going to be different. So unless the force is exactly the same from the spring as the motor, then you're going to have a phenomenon that causes a difference in cadence. So what that means is that the motor will be going at one speed, but the speed of the retraction and insertion is going to be of the needle head itself in these uncoupled devices will be a combination of the speed of the motor pushing in and the force and speed of the spring pulling back. And so because it's unlikely that the force is equal between the spring and the motor itself, you're going to have an incomplete or an irregular retraction. Now, if you're some devices may try to fix that by actually making the force of the spring higher so that it's pulling back with great force in order to make sure that anytime a piston pulls back, the spring also pulls back at the same cadence. But the problem with that is that it's tricky, because if you don't get the spring to be exactly the right force, then basically you're going to actually make the motor push harder, and you're going to end up with an incomplete insertion. So even though you try to put your device to go to a certain depth, it's always going to be off slightly with calibration. Now, also, the other component of this is that springs and rubber and such, with repeated use of that mechanism, can lose its elastic properties. So you have to make sure you're using a very high quality, either metal or rubber, in order to make sure that the retraction force is consistent throughout the entire use of that particular disposable. And so there's a lot of considerations and variables when you're using an uncoupled mechanism. And that's one of the main reasons why we engineered the SkinPen Precision to use a coupled mechanism. It takes those variables out of commission, out of the equation. And so if you look at the next slide here, what we did is this is the first part of our show and tell, is that we actually took several devices, and right here we have 3 of them. Our device is on the most left-hand side, and then we have brand Y in the middle and brand Z on the other end. So if we, Angela, if you go ahead and play the video here. Okay, so we did it at higher speed. Now it's in slow motion. So this is very much slowed down so we can capture the reciprocations. And what you can see here is this is a device that uses a rubber spring, it's an uncoupled retraction. And so you can see that what's happening is you're getting a bit of a cadence of where it's not completely retracting the needles. So you can see a couple of insertions where it's not retracting completely. Let's see if we can start it again. Okay, so basically, there you go. So you can see where it's got a couple cadences where it's not retracting completely, and then it retracts completely. and a couple of cadences where it's not retracting completely and then it's retracting completely. And so we have an inconsistency of the retractions. Now, this particular device, we'll look at the depth of penetration as well later, and you'll see that also there's issues with the consistency of penetration based on the calibration of where the depth we're trying to achieve. But if we look at the middle device here, we'll see that this particular device has a different type of issue. So this particular spring coupled retraction actually doesn't even fully retract. So it's got a pretty consistent cadence, but the problem is that, can you go ahead and play it again, please? The problem is that it's not able to fully retract. So as you can see, the needles are going to be constantly in the skin. So if you drag that across the skin, you're going to end up causing further injuries and tears. So if you look at the SkinPen Precision, we did the same thing here, and you can see that with a coupled retraction, you're able to get a complete retraction every time. And so that is actually a proof of concept type of set of videos here to show you that the difference between the spring modulated retraction and the coupled retraction is quite significant when you're looking at the quality of the device and what you're trying to achieve. So on some of them, for instance, brand Z, you may not worry as much because there is going to be complete retraction. It's just going to be inconsistent. Where with brand Y, and we'll describe it a little bit later, you actually see that you're not even getting any complete retractions, or the SkinPen, you're always getting complete retractions. So if you go to the next slide here, you can see what we decided to do is look at the depth of penetration. Now, this is a study that we did in collaboration with the University of Texas Arlington, and so it was in the lab of Dr. Ki-Tai Nguyen, and her graduate students also assisted with this as well, her postdoc and graduate students. And then some of our scientists, as well as Dr. Brian Jones, who heads up our medical affairs and clinical ops group, he participated in this as well. And so this was kind of driven by a publication in 2017 where Dr. Gordon Sasaki, and I won't talk about the actual devices that were measured in this particular study, but this is in the journal article as cited on the bottom right-hand side here, so you can look it up yourself if you're interested. But he looked at a particular device, and Gordon does really great work. Dr. Sasaki, I always enjoy his publications. He does very thorough studies usually. And so you can see here that he dialed the device to specific lengths starting at 0.25 millimeters up to 2.5 millimeters. And this particular device, not the SkinPen Precision, but this particular device, as you can see, when he measured the depths of his microchannels that were created, they were pretty consistent up to about 1 millimeter. And then at 1 millimeter, you see that there's really not a significant change. On average, they're still at about 1 millimeter to 1.25 millimeters, even if you go up to upwards of 2.5 millimeters. So there's an inconsistency, and some of that is probably attributed to the actual mechanism by which the motor translates the energy into linear motion. So we decided with the University of Texas Arlington to go ahead and set up some apparatus so that we could measure using skin substitutes how deep the actual needles penetrated in this gel that is a tissue substitute. And so you can see here on the left-hand side, they made sure that the gel was homogeneous so that every time you're looking at it, you're going to see the true measure of the depth of the penetration of the needles. And then on the right-hand side here, that's how we were setting up all of these devices through a mechanism that allowed us to make sure that there's no human error introduced here as far as amount of pressure, etc. So they were all lowered to the surface of the gel substitute, and then they were ran, and we measured the depth of the penetration looking at it through dyes that were entered into the gel. As you can see here, this is the measurement of the SkinPen Precision, the results. So we ran it in triplicate, and you can see basically, we'll just go to the next slide here, which is the graphical representation, or the graphical representation here. And you can see that it pretty much measured up. So when you dialed it to 0.5 millimeter, you got 0.5 millimeter, give or take a small variation. The 1 millimeter seemed to be a little more variable there, but on average it was around 1 millimeter. And you can again and again up to 2.25 millimeter they measured. It was the final measurement here. And you can see they all tracked pretty close with very, very tight variation. So the standard deviation was not much for most of these measurements. However, what we decided to do was then compare to other devices, because we wanted to see, okay, well, our device, stacks up pretty well. We know that when we measure it, we dial to a certain needle depth, it usually penetrates to that depth. It was validated with the FDA, and here we're showing you today that independently, when we do measurements through a third party, we see again that the measurements do penetrate to where we dial them. So here we looked at all these devices and we measured them to around, we set them to the device to around 2 millimeters. And what we found was the SkinPen device, as we saw before, went to about 2 millimeters, give or take a small variation, as you can see here. Then we went to brand X. So brand X, we set at 2 millimeters, and we saw after we did 5 runs of this particular test for this device, and you can see that although we set it at 2 millimeters, consistently we saw around 1.75 on average millimeters. So it's calibrated about a quarter less millimeter than what we're seeing. Brand Y, when we set to 2 millimeters, we actually saw that it went to about the same, about 1.75 millimeters on average. And then brand Z, interestingly enough, brand Z is only going to about 1.25 on average. So although set upwards of near 2 millimeters, I believe this particular one was actually at 1.9. It was set to, but it actually went to only 1.25 on average. So if you look at comparison of all these devices, you can see that this is just a direct comparison of the slides I just showed you previously, that brand X, Y, and Z do have a variability from where they are actually set to. So while they have a pretty, you can see brand Z and Y, their standard deviation is about 0.05, and while that's larger than the brand X and SkinPen, which is 0.03 and 0.02, it's still not that bad as far as the variability of the actual penetrations. However, they are off. The brand Z is the most off from any other of the devices we tested, at about 3/4 of a millimeter off from where it was set, where brand Y and brand X were about 1/4 of a millimeter off from where it was set, and SkinPen was hardly, it was not off. It had a normal variation of less than 10%. So here's a graphical representation of that. Again, what the considerations that are interesting here is that the brand X is actually a leading brand outside the United States. It's not really found in the United States, but it's outside the United States, a leading brand. And interestingly enough, brand Y that is comparable to brand X is not a leading brand, but is something that is a brand that is something that was found for very cheap on Amazon. So it was a very low quality device that, well, we're expecting for the price that we bought it that it's low quality simply because of it was under $100. Yet it had a same calibration as brand X, which is a much more expensive leading brand outside the United States. And then brand Z is actually one of the other FDA cleared brands. And as you can see, the calibration was off the most of any of the other devices tested. And now, of course, we have, I think at this point, 4 FDA cleared devices. And we won't go into actually which device it was, but you can see that even, again, just because something uses the moniker of microneedling, just because it has a validation through a regulatory body, does not mean it necessarily has quality. All right, now we're going to move on to fluid ingress protection. So basically, when we did our work with the FDA several years ago, what we were asked to do by the FDA in order for them to feel comfortable with us moving forward and getting our clearance was to show a worst case real world scenario. So we actually went above and beyond. So in a real world scenario, you're going to be treating the skin and having fluids on the skin, and you're going to have an array of microbes from the microbiome of your skin. So for instance, we have here Staphylococcus epidermidis, we have some Bacillus subtilis, which is in a very low portion. We have Staphylococcus aureus, which is known to be associated with pathogenicity. However, it's also in a very little amount on the skin typically. and then E. coli, which can be associated very little with the skin, but mostly can be contamination from the gut as well as contaminated food sources. And so we actually took a soup of these bacteria and we put them into high quantities. So down at the bottom, you can see that what we did is we actually used a soup where there was per milliliter 125 million bacteria per milliliter. So that's of course going to be much higher than you would see in an actual treatment. And then also when we did the study, which we're going to show you a video in a little bit, which shows you a technique we looked for prevention of fluid ingress. I'm not going to show you right away, but it's exactly the technique we used, where we took the soup of bacteria and we ran the SkinPen on that soup of bacteria for 45 minutes. Every 5 minutes, we would turn the SkinPen upside down. We would change the setting to the next depth, turn it back right side up, and then we would treat for another 5 minutes. So it was in 5-minute increments, turn, switch, and treat again for a consistent 45 minutes. And what we saw was the next slide. Well, the next slide is what we did. We actually, after we did that test, we swabbed the inner part of the device. So the inside, the coupling mechanism, and then inside of the disposable tip as well with a cotton swab. And then we actually swabbed that onto Petri dishes to show that there was no contamination. Now, the idea is that when you do that, any one particular bacteria that grows onto that Petri dish will form a colony. So you'll see a little white dot, a little white mound called a colony. And that colony represents one single bacteria that multiplied and became many, many bacteria. And so as you can see here, when we tested before, we saw ND, which means no colonies observed on either the pen or the cartridge. So we expect that because we sanitize the device prior to using it. And then after treatment, you can see here that there was no colonies detected as well, none detected. So you might say, okay, well, how do we know that this was working? So if we go to the next slide here, which is the positive control. So we took that same soup, we diluted it 1,000 times, and the top left-hand corner is when we plated that dilution of 1,000 times is what we would see. We call that a lawn, where you're not able to distinguish the individual colonies. Then you can see we did a serial dilution where we diluted it next 10,000 times, 100,000 times, 1,000,000 times, and then 10 million times, which is on the far right here. And you can see once when we diluted it that much, the positive control only shows one colony. So basically, if any bacteria are able to get through the device into the mechanism, even in a real world worst case scenario, we should be able to measure that. So as you can see in the next slide here, this is before. So when we swabbed the device before, we saw that each of the cartridges do not have a colony that is formed. Now, what I'll show you is that you can see a little dot on the right-hand bottom Petri dish. That is not a colony. Those are bubbles that form when you're pouring the agar gel. So if you, Angela, if you go up the slide before, you can see a very big difference between a true colony, as you can see there, and then if you go back down to the bubble. So I just want to show you that in case that confused anybody. And then if we go to the next one, we look at the pen, where we actually sampled each of the pens before we did the test, and you can see again, nothing on those plates. Then after, which is the next slide, we can see that when we looked at the pens after, the cartridges after, we didn't see not even one colony. And then the pen itself afterwards, we did not see even one colony. Okay, so all of you might say, well, this is great. It convinced the FDA, right? So that might be good enough for me. Other of you might say, well, you know what? There's people coming into my practice and they're actually pouring liquid into these different cartridges and showing me that there's fluid flowing out when I'm pouring copious amounts of fluid into these cartridges. So therefore, I'm skeptical. Well, we'll have the answer for you in a second. But before we do that, we're going to look at this 3-day experiment that we did with the sheaths. And so another sense of security when we use our devices is making sure that we're, even if we are going to wipe the device down after we use it, we want to make sure that we're not introducing enough to actually get into the nooks and crannies. So one of the other things that we did when we designed our devices, we ultrasonically sealed the handpiece so that if you were touching it, even without a sheath on it, there's not a lot of nooks and crannies for any bacterial or virus or anything like that to hide. That being said, there's no need to take chances, and also we don't want to make life harder for everybody, so we have the sheath to actually protect the device. And so what we were able to show is that if you look at the sheath here on this next slide, you can see how the sheath is attached to the device. And actually, can you go a couple slides up? You need to go up. There you go. And so you can see that basically when the sheath is attached to the device, that you have the disposable part of the device, and then you also have the sheath covering the non-disposable portion. And so the part that people want to know is that, okay, if I'm using this device and I get contaminant onto the biosleeve or the biosheath or onto the cartridge itself, will I be able to make sure that the device itself is not in contact with that potential pathogen? And so what we did was we took that same soup that we talked about before and we literally sprayed it and poured it on top of the biosheath and the cartridge. And the next slide here is exactly what we saw. So not what we saw, but this is the positive control again. So this is taking that same soup and growing it on Petri dishes to show that what happens when you see a serial dilution of those bacteria. And then after the next slide is before each of the experiments, you can see the first pen, we measured the actual metal of the coupler mechanism, the plastic of the actual outside of the actual device, and then the inside of the sheath. And so we did that with pen 1, 2, and then the next slide is pen 3. As you can see, there was no contaminant on any of these. Okay, this is not, it's the slide before, so. Okay, so this is pen 3. So you can see there's no contamination. And if we go to the next slide, this is actually the outside of the sheaths. So you can see on the outside there was a whole lot of bacterial contamination, yet inside the sheath we did not even see one. And so it basically is inhibiting even the smallest microbes from getting inside to the actual pen mechanisms. And then here is the metal part of pens after treatments. This is the plastic part of the pen, and then the cartridge. So again, let's talk about what we discussed earlier about people that have decided they wanted to take it upon themselves to measure whether or not pens can allow for or can mitigate fluid ingress. And so we've had people, there was a person in the UK that actually decided they wanted to go around testing cartridges by taking that cartridge and pouring liquid into the cartridge and then seeing whether or not it flowed out of the cartridge. Now, some devices actually try to mitigate cross-contamination by putting a physical barrier inside the disposable itself so that nothing can actually flow through. The problem is that if you're going to do that, it's going to have to be an uncoupled mechanism. And so it's very difficult to have a coupled mechanism if you're going to have that type of a seal there. So we designed ours a separate way. So if you actually look at the device, we can go ahead and populate the slide here. So if you actually look at the actual mechanism, if you look at the device, I can take off the top here. So you can see that the top portion here is not sealed onto the bottom portion here. It was never intended to be. So this part here, so if you poured liquid into the top here, because it's not sealed, if it's enough liquid, it's going to get past these threads and actually flow out of the cartridge here, at this part here, not down here. So as you can see here in this schematic here is what I'm trying to kind of explain to you, is that you can see that the fluid, if it came through the device, which it's unlikely to do. So if you look at the bottom part here, at the very bottom, you can see through the actual diagram here that we built in vents, just like most devices. They have vents, they have anti-suction cutouts that cause a scalloped edge. These are all meant to be there so that they don't cause suction. So you're not physically sucking fluids into the device. Now, that being said, it is possible that when you turn the device over to change settings and such, that fluid could flow down towards the device, because now you don't need suction, you have gravity pulling it down. Most times when you're doing treatments, you don't have copious amounts of fluid, but if you're using something like PRP or something like that, that might have more fluid than other types of treatments. So that being said, how do we know that if it's flowing through, it's not actually getting into the inner workings of the device? Well, First thing I would tell you is if you looked at what I just showed you with the bacterial contamination, we saw that not even one small bacteria was able to get through the device into the actual pen. But we thought we would show you on a more macroscopic, more graphic level how this actually works. So this next slide here is actually a video. So in a second, when I stop talking, you can go ahead and toggle to look at the video full screen if you're on a phone or on a computer. And so my medical affairs and clinical group put this together where they actually used cow blood. So if that helps you, it's not actually human blood, it's cow blood. And so they used copious amounts of cow blood and they actually poured it into the device. And I want to show you why exactly, even if it does get through this portion here, this top portion here, why that is not an issue. So Angela, if you can go ahead and play. Okay, we're not hearing any sound, so let's start that over. And so while she's doing that, before she starts it again, I want to mention that this first video we did not cut because we wanted you to see that from start to finish that we did not mess with the pen, that it actually truly shows that if it ingresses through that top portion, it's not going to ingress into the actual device. And so the second video we cut because for sake of time, we didn't want you to sit through a 20-minute video. So this first video is about 4.5 minutes, and the second one's about 2.5 minutes. So Angela, if you can go ahead.
To address the question regarding the potential backflow of fluids by the SkinPen cartridge into the SkinPen hand unit, we have made 2 demonstrational videos that we believe support the laboratory studies that were previously submitted, reviewed, and gained clearance for the SkinPen by the US FDA. In the first demonstration, we will show that using cow's blood as an example of biological fluid, the SkinPen cartridge is designed to limit the ingress of fluid into the hand unit drive shaft and cartridge coupler area, which we are referring to as the inner chamber. With the disposable one-time use cartridge being attached to the hand unit covered in the protective biosheath provided with the cartridge kit, the unit is set up standing upright. Now again, this is essentially opposite to how the device would be used on a patient, but this is to exaggerate the exposure conditions seen in a clinical practice. As cow's blood is pipetted into the opening of the cartridge, we have added much more fluid than would ever be seen in actual operation. As you can see, it nearly fills the upper open chamber area of the cartridge. After a few moments, the blood is able to pass by through the outer plastic containing the depth adjustment threads and outward to the biosheath covering the hand unit. Clearly, this is an excess of fluid, as well as the help of gravity to cause the flaw by the threads and out across the hand unit. Once the fluid has moved from the outer chamber, we are able to clean the hand unit biosheath with a couple of sani-cloths per the use instructions. Once clean, we can remove the cartridge from the hand unit. When apart, it can easily be seen that even with an abundance of blood introduced into the upper opening, no blood reaches into the inner chamber area, as both the drive shaft and cartridge coupler areas are free of blood, further demonstrated by using a cotton swab to show no movement of fluid into the connection area.
So as you all can imagine, if you were to get blood on a cotton swab, it would be very visible. The vermilion would be very visible. So I think that that's an excellent demonstration to corroborate what we saw with the microbiology experiment that we did a couple of years ago with the FDA. This next video is basically answering the question, well, Thomas, you know, when you did that experiment, it was not operating, meaning the reciprocating mechanism was not going on and off. And so what we did was what I mentioned earlier, we redid the microbiology experiments, but not with bacteria. We did it with red food dye so that we could actually see that if we're running the device for 10 minutes straight upside down, whether there would be ingress into the device versus just through the flow of the top portion of the device, as we saw just a few minutes ago. So this next, I don't think this is it. So go to the next slide. Okay. This one will be 2.5 minutes. There will be some cuts. So again, this was for sake of time. So we'll discuss it afterwards. Go ahead.
In the second demonstration, we have created an exaggerated use experience using a plate that contains an artificial skin to which an abundance of red liquid, food colorant, has been placed. Placed on the tissue and liquid is SkinPen Lift, which is provided with the cartridge kit and acts as a glide in performing the microneedling procedure. The unit is turned on and microneedling of the tissue in the liquid is started. As you can see, there is considerable amount of liquid on the plate, an amount that would never be seen in real practice. The liquid fills the lower cartridge area entirely and even dissipates liquid through the cartridge tip vent holes. Remember, this is for an exaggerated demonstration regarding liquid exposure to the SkinPen cartridge. After a few minutes of microneedling in the liquid, the SkinPen is left running and tipped upside down, thus allowing any fluid in the lower cartridge area to remain in the cartridge. The unit is allowed to remain on. After 10 minutes of constant running, the unit is turned off and the cartridge and the hand unit are separated. Again showing the inner hand unit drive shaft and cartridge coupler inner area is free of liquid. Further wiping with cotton swabs demonstrates no ingress of liquid into this inner chamber area.
All right, so again, I think that one of the takeaways from this is that, you know, from what I said from the very beginning in this talk is just because somebody uses a moniker of microneedling does not mean that all devices are the same. that they don't work the same, that they're not designed the same. And one of the case in points is this particular thing, that our cartridges are definitely not the same as others on the market, as we discussed today. The fluid ingress here, you cannot test ours the same as you would test another, simply because of the way it's designed and engineered, is that you have to do testing that is validated, that is standard, and that is in context of the device itself. And so that being said, we're running over time, so I'm going to stop there, but we have a lot more to talk about that's very interesting on the handpiece next portion of this webinar, which will happen, I'm not sure what date yet, but we're right now getting ready to do that one. I want to say thank you to Dr. Brian Jones and Dr. Mona Alcom, who did those videos for us. And hopefully we'll have a few more videos and show and tell experiments for you next time as well. So with that, I'm going to show you this last slide here that has my contact info. So I have my Instagram moniker on there that I would say, please do follow me, because I'm gonna in the future have a lot more, not just on SkinPen, but we have some very exciting microbiome technology coming out this year. And we're gonna have a webinar series coming out monthly from here till next year that I would think you would very much like to watch. And so go ahead and follow me so you can see when those will be. If you have questions on today's lecture, send those to midinfo@crownaesthetics.com.